Kia ora koutou, ko perepere te maanga, ko waitohi te awa, ko tokomauro te waka, ko roa kawa kawa te moana, ko te Aotearoa me nai tahu nai iwi, ko waikawa te marae, ko Nathan Kenny taku ingoa, no reira, tēnā koutou, tēnā ora tātou katoa. Thank you very much for coming along. My name is Nathan Kenny. I am originally from up around Te Tau Ihu, not far from here, Picton. I've got familial links all around there, but I am now, as Peter says, down in Otago. And today I will be telling you about work that I have been doing in the course of my last few postdocs before I came back here on the freshwater sponge, Ephydatia muelleri, which is, as well as being astonishingly beautiful, as you can see from this fried egg looking structure in front of you, is a really useful critter for all sorts of things going forward. You don't find Ephydatia in New Zealand, but you do find sponges. Sponges are astonishingly prevalent, even if you might not realize it. So just behind here, there is a lovely river, and in amongst all of the shopping trolleys running upstream to spawn and all of the rusty bicycles and everything else, there are undoubtedly freshwater sponges. If you go out into the sea, a large part of the benthos, including in places that you wouldn't expect to find much life, like the deep sea or in Antarctic seas, is made up of sponges. And they can form all sorts of beautiful arrangements, as you can see on this plate from Heikel on the left. And for a long time, sponges were a bit difficult for people to place. They don't move much when they're adults, and they have a bit of a mix of phenotypic features. And people weren't quite sure where to put them. And if we look at this plate of Heikel over here, up the top in the middle is where he popped the sponges with the protists, which is quite wrong. And for a long time, people were debating, okay, are sponges a kind of plant? They don't move. Are they a mineral? They're quite hard. Are they animals? They move around when they're juveniles. They have a whole lot of features that we do find in animals. And the answer is yes, they are animals. They are pretty clearly animals when you look at the embryology. And now that we live in the day of DNA research, it's abundantly clear that they're animals. They have a few features that make them quite special, though. So despite their simple body plan, where they're built around these inhalant canals, they bring in water, they take up all the things that are in the water. They can remove pretty much everything in water that they take up. It's quite astonishing. And they pump out the larger holes that you see in this sponge here are the exhalant channels. So that's where water is coming out. They pump out water in large volume. So quite a simple body plan where they sort of grow quite in a sort of mat across wherever they're growing. They have few real tissues as a result of this. They don't have nervous systems or muscles or things like that that we would think of as tissues, but they are nevertheless genomically very representative of animals. They have a very complex genome. They have more genes than you find in your average animal, actually. So in a sponge, you would expect to find about 40,000 genes, as I'll talk about more in a second. But in us, you find about 25,000. In Drosophila, you find about 20,000. There are other species of animal which have far fewer genes. So despite the fact that they've got a very simple body plan, they possess all the gubbins, all the stuff that makes an animal an animal. And that's what I'll be talking about today. So I noted that sponges are unambiguously animals, but there is an ongoing and even though it feels like quite an angels dancing on the head of a pin, quite esoteric debate, it's vitally important that we come to the bottom of it, sort of investigation into where exactly sponges sit in the animal tree of life. So the big controversy is over whether ctenophores, which are sort of like disco jellyfish, they're very pretty, largely see-through, but they do have these lovely teens, these sparkly bits that enable them to attract prey and move around. So maybe they are the outgroup to all other animals, or maybe sponges are the outgroup to all other animals. And this is quite a fraught scientific debate. And the reason it's quite fraught is if you know what the common ancestor to all other animals look like, you can begin to infer what the characteristics that the most ancient animals possessed. So If we look at a tree of life, something like this, we know that animals came about somewhere down here and had some traits, but what we don't know is whether they had things like a nervous system. The common ancestor of animals may have had a nervous system that was lost in sponges, or more likely, it might have had a nervous system that evolved further on down the I don't want to say chain of being, but across the metazoan stem lineage as crown group animals came into being. So by knowing what is the outgroup to all other animals, you can begin to ask questions about what the earliest animals looked like. And that's why it's really important for us to understand sponges, understand ctenophores as well, if they happen to be the outgroup to all other animal species, and be able to work out, okay, how did animals evolve? What were the special traits that gave rise to animals and enabled them to, well, take on more or less sophisticated forms, as you can see here. And so there's all sorts of things that make animals special. Part of that is in the genome. And we started out this project thinking that if we had a good sponge genome, we'd be able to dig into these things and work out what makes animals special, what makes animals tick. So we weren't the first people to look at sponge genomes. There have been a number of attempts to sequence sponges over the years. This has been difficult both from a technological point of view. As you all know, genomic sequencing has been advancing at a rate of knots in the last few years, but also because sponges tend to have lots of other things in them. Sponges are quite famously quite full of holes, and all sorts of animals like to live in those holes. They also have a lot of bacteria and other single-celled organisms that will grow inside them. And there's quite a lot of complex symbiotic relationships between sponges and the things that live inside them. So it's been historically quite difficult to sequence sponge genomes, and I'll get back to how we got around this. Despite this, people have made heroic efforts to sequence sponge genomes. The most famous of these efforts is from about 10 years ago, and is the Amphimedon queenslandica genome. This is a sponge that is found around Queensland in the north of Australia. And the Dignan lab over there did a wonderful series of work digging into the gene complements of the sponge. And from this, we know full well going in that sponges have a large gene cassette, about 40,000 genes, and an extensive gene cassette that is quite common to that found in us, in Drosophila, and any other metazoan you care to go and look at. But it is missing some key features. Along with that, we know from sponge genomes that we've looked at in the past that they have quite compact genomes. So Amphimedon queenslandica has a genome size of about 166 megabases, contrasted with our genomes, which is about 3.2 gigabases, quite a step change in difference. And along with this, they have some genomic features that we know already. So smaller intron size, there's a little bit of alternative splicing, but not that much. And we also know that they've got a highly methylated genome. People have looked into this in the past. Just like vertebrates, they use DNA methylation, likely to silence genes in much the same epigenetic pathways that we do. But despite this, we have no well-scaffolded resource. So the Amphimedon genome was state-of-the-art 10 years ago. It's no longer state-of-the-art. Nonetheless, it was very useful. So inferring ancestral characters from sparse resources is never easy. So there's the saying, absence of evidence is never evidence of absence. Without a really good genome, if you don't find something, you don't know whether it's not there because you haven't found it in your slightly fragmented genome, or whether it's really not there. So having a really good assembled genome resource is always important. And the species that are sequenced aren't necessarily those that are easy to work on. So Amphimedon, like I said, is found in the north of Australia, but getting them to grow in Dunedin is always going to be a difficult task. So I'd like to introduce you to Ephydatia muelleri, which is the sponge that we worked on. Ephydatia is found across the northern hemisphere. So in green on that map that you can see down the bottom left are all the places that it's known to be found, as been described. It is almost certainly been found in, found in other places. It's just that there's been no clear scientific description. And it can move around on waterfowl. So a characteristic of freshwater sponges, all freshwater sponges except those found in Lake Baikal, have these white dots that you see in here. These are called gemmules. what gemmules are these cysts of cells where there's a hard outer layer, and inside there's pluripotent, so cells that can grow into a complete sponge once they're given the chance to do so. These gemmules can survive really harsh conditions. They can survive freezing, they can survive desiccation, they can survive high heat. And they're the things that allow freshwater sponges to survive when things like waterways dry up, but also to spread, because if you can get on the foot of a duck, you can fly quite happily on the foot of that duck wherever that duck goes. So the modern freshwater sponge radiation isn't all that deep. Dates back to about 32 million years ago. Ephydatia is one of those sponges. And all of these sponges have got all around the world, including here in New Zealand. There's freshwater sponges, not Ephydatia unless it's been carried in on something. I doubt a duck could go that far, but maybe a sooty shearwater or something might have picked one up. But there are other species found in all sorts of nice places. Now, another nice thing about Ephydatia, which isn't necessarily true of all sponges, is it can reproduce asexually via the gemmule method that I talked about before, but it also retains the ability to reproduce sexually. So if you are nasty enough to Ephydatia, it will create eggs and sperm. It will hold on to the eggs. It will release its sperm into the ecosystem. Sponges are remarkable. They can tell their own sperm from any other sperm. They'll digest everything else, sequester sperm from their own species, move it to the eggs inside them, fertilize those eggs, keep them inside for a little while, and then release what's called a planular stage larvae, which is free swimming and really gives away the animal characteristics of sponges, which then will turn into an adult, and then you can either have sexual reproduction or asexual reproduction. That's really important because asexual only species tend to be quite weird genomically because they're trapped into evolutionary pathways that might not be representative, and they don't have the ability to shuffle genes like sexual reproduction allows. So having those dual pathways to survival means that this is a really useful genome to look at. The other useful thing about Ephydatia and other freshwater sponges, as I've sort of intimated already, is these gemmules, which allow you to freeze, if not hundreds, then thousands of individuals per sponge. You can freeze them at -80 degrees. You can send them to your friends on the other side of the planet, who will then unthaw them, put them in some fresh water, and allow them to grow and develop on a plate. This means that you can have your own samples that are identical genetically to those that your mate in Japan or in Canada is working on, and you can use them for your own purposes, which is fantastic if you're thinking about all the other sponges we've used before, which would require you to go to an island off the coast of Queensland in Australia, or sample in a more ad hoc basis. So very, very useful. And this was also really, really useful when we started doing our genomic DNA extractions for reasons that I'll get to in a second. So the sponges in particular that I'm working on, and all of this work is based on work that was driven forward by a lady called Shelley Lays, who I'll talk more about towards the end of this talk. These sponges were gathered from what's called the Kapoor Tunnel, which is in Victoria in Canada, which is a ginormous water chute. So 99% of the year, the tunnel that you can see in this photo would be filled 3 times the height of these people here, filled with fresh water running down to feed a number of cities. Sponges obviously like this environment. There's water going past all the time that allows them to filter feed and eat. But you'll notice that these sponges, in contrast to some of the pictures that I showed earlier, these sponges are quite white. So here's another series of pictures. These sponges do have some symbionts inside of them, but they don't have algae. So if you can imagine that historically it's been difficult to extract DNA from sponges because they've got lots of symbionts. Here we have sponges that don't have algae at least. And if you take the gemmules and if you allow the gemmules to grow in an aseptic environment, all of a sudden you can get DNA from something that A, doesn't have the algae that you'll find in other sponges, but also has a much lower amount of those symbionts, which means that you can get a lot more good DNA. And that's what Sally and co did. And that's where I stepped in. So from this, we went forward with genomic DNA sequencing Obviously, for a genome, you need to sequence somehow, and then you need to put it together. So the approach we used, which is quite a nice one, is using PacBio sequencing to initially create contigs. So we did some PacBio sequencing, and then we used the dovetail approach, which involves two kinds of high-C library. High-C libraries give you long-range interaction data. So the way you make a high-C library is you take your cells that you're going to make the genome from, you cross-link the DNA inside, so all the DNA that was close inside each of those cells is now linked to the things that were close to it. You cut out the DNA, and then you, via a variety of steps, send it forward for sequencing. That means that DNA that is in close proximity, you are more likely to find reads of, which means that A, you know which DNA is touching each other, and B, you can infer longer range regulatory links, which is important, and I'll get back to in a second. So through this, we ended up with quite a nice genome. So here is the example of Ephydatia here, alongside Amphimedon queenslandica, which is the previous best sponge genome that I was talking about before, and two other invertebrate genomes that are of quite good quality. And you can see that our genome size is about twice that of Amphimedon. It's not unusual to get a bit of variation in genome size, as you'll all know. But what we've got is far fewer scaffolds, and even this is a little bit of a misnomer, as I'll get to in the next slide, because we've got some pretty clear chromosomal scaffolds, as I'll talk to in a second. We've got a much higher N50, so this is a much bigger genome in terms of the scaffolding than almost all other genomes that you look at, and certainly much, much better than the sponge genomes that have been around today. And in terms of gene models, again, we see that we've got a very complex genome in terms of number of genes. Nearly twice the number of genes that you'll find in most invertebrates, and up there with Amphimedon. So this is definitely a shared sponge character. Now, I mentioned before how we had slightly over 1,400 scaffolds, but we had quite a few chromosomal length scaffolds. So almost all of our assembly is on 24 of the scaffolds. In excess of, I believe, 87% of the genome is in 24 scaffolds, corresponding to the 23 chromosomes of the species. So we've had to be very careful not to call these, number these unambiguously as chromosomes, because two of these ones will be joined at the centromere and will be another whole chromosome. So 24 into 23, you join two of them together, all of a sudden you've got 23 chromosomes, but we don't know for sure which one that is. But nevertheless, we've got a very good assembly and can infer for the most part what the chromosomes in this species are. Just to convince you of how much better this assembly is than other sponge resources to date, here we've got a representation of the genome where the size of each of these boxes represents the size of the scaffold. The size of the total box out here is the size of the genome as a whole. And the number of boxes that you see inside there are the number of scaffolds. And I think it's quite clear to the eye that this is a much better scaffolded genome than any of the previous best sponge genomes that have gone before. So hopefully I've convinced you that this is a good genome and useful to be using. And we can start to ask questions about how sponge genomes relate to those from other metazoans. So sponge genomes are generally much smaller than other metazoan genomes, but what allows them to be smaller? So here we've got a comparison of exon, intron, and intergenic lengths, where Ephydatia and Amphimedon, so the two sponge samples, are in green and pink respectively. And you can see that while exon length is pretty much identical between sponges and other metazoan genomes, the introns and the intergenic length is much, much smaller in sponges than it is in other metazoan genomes. So the way that sponges are getting more genes into a genome is by cramming them all together. So the actual genes themselves don't change. The coding structure of the genes is not that much different. It's the bits in between them. Another way you can begin to understand what's in a genome is looking at the annotation. So looking at what gene identities you've got inside there. So if we take all of our genes from Ephydatia, blast them against everything in the NR database, the NCBI NR database, we had started with about 40,000 genes. About 2/3 of them have a hit, that means that we've got a sizable amount of dark matter, so about 10,000 genes, which we can't firmly annotate to a function, a pre-described identity that is seen in other species. And this means that there's lots of scope for this sponge to be doing interesting things that I'll get to in a second. We can use this resource to examine well-known gene pathways and see what's present in sponges. All of a sudden, we can begin to make those presence-absence arguments that were difficult when we had a poorly scaffolded sponge resource. And for example, Wnt signaling is well known in lots of contexts as a developmental signaling pathway. And if we look at these canonical pathways, all of the core genes tend to be present, shown in green in sponges and in Ephydatia in particular. Orange genes are absent from Ephydatia, but not necessarily expected. So some of these genes are known to be specific to particular organisms. They're just included here out of completeness. But the important thing I want to take out of this is the core pathways are present in our genome. This is a good genome and a good resource to be working with going forward. So compared to the wider metazoa, important points to remember: sponges have small genomes, more genes, but much the same pathways, and a few of the genes have unknown functions. So Ephydatia is no exception in that regard. So thinking about that question that I was talking about earlier, how does the genome help us to understand what makes animals special? So you may have heard of this concept, macrosynteny, where genes are carried in sort of bags of genes, chromosomes being the bags of genes, where the exact order on a chromosome can get shuffled over time, but the genes are held in a relatively conserved relationship. And this is an example of this. So this is Ephydatia compared to Trichoplex adherens, which is a placozoan. And this dot plot represents the Ephydatia genome on this side, the Trichoplex genome on this side. And where you get a bidirectional best blast hit of a gene, we've put one green dot. So where you get lots of green dots in close proximity is where genes are. So in both Trichoplex and in Ephydatia, all held on similar common chromosomal loci. So they might change in order, but the genes that are on those chromosomes are being held all together. And so this is one particular example that's boxed in red here. You can see that there's lots of genes in Ephydatia scaffold 22, lots of genes in Trichoplex adherens scaffold 5 that are on what are probably historically or ancestrally homologous chromosomes. The order has changed, but the genes are still all together as one unit. Another thing that we were able to get out of our Hi-C sequencing data, as I've sort of alluded to already, is evidence of chromatin structure. So Hi-C gives you long-range interaction data. So in these plots, the more red that you see, the more crossing over there is between a chromosomal location shown here and a chromosomal location shown here on the same scaffold. So in some metazoans, you see these structures called topologically associated domains, TADs, which are in red that you can see here. These indicate that within these units, you've got areas of the genome that are influencing other areas of the genome across quite a reasonable chunk of this genome. So if there was no long-range interaction, what you just see is a red line running down the middle. So we also see these topologically associated domains in sponges. This is what you see in mice, what you see in Drosophila. You don't really see it in Arabidopsis, although you do see it in some other plants. And you can sometimes see it in bacteria, although not to the same extent. What we don't see in sponges is in vertebrates, you get these loop structures that are found at the corner of these topologically associated domains, which are normally associated with CTCF binding sites. We don't see these at the corners of the topologically associated domain in sponges. They're in all sorts of off-center locations. So it's almost certain that these are not playing the same role in sponges as they are in vertebrates. We were also able to look at DNA methylation, which is normally a signal of gene silencing. In vertebrates, it's widely known that there's relatively high levels of DNA methylation when genes aren't turned on. In invertebrates, that's not the case. Different invertebrates can have quite low levels of methylation across their genome, only methylating particular sites via CpG mechanisms. All sponges have reasonably high levels of methylation. So here's 3 sponges here, Amphimedon that we've talked about, Ephydatia and Psychon. Ephydatia doesn't have the really high levels that we see in other sponges, but it is more like vertebrates than we see from other invertebrates. So this DNA methylation as a means of gene silencing is probably something that is ancestrally shared. So just to run over those facts again, very briefly, macrosynteny is conserved across animals. The common ancestor of animals could likely organize chromatin in topologically associated domains. Sponges have vertebrate-like methylation patterns, and so there are some characteristics of the genome of animals, from sponge to human or sponge to you name it, platyhelminths, rotifer, whatever you're working on, that are shared across the animal tree of life. So another thing that we're able to do with the genome is start to dig into sponge development. We've got a lot of raw material here that we can understand, use to understand how sponges go about their business. And there's nothing more complex than growing a whole new organism from a single cell. So I've already mentioned this, so I won't dwell on it. Ephydatia muelleri can reproduce both sexually and asexually. So both those options are open to it. But here we've just focused on what happens 99% of the time, which is asexual reproduction. So here we have triplicate samples from stage 1. So Jimmy was just starting to grow through to stage 5 when you've got a small but pretty well-formed sponge. And what you see is more and more methozoan-like transcription happening. So this will become apparent when I show you the next slide. There are genes that are special and specific to stage 1, genes that are turned on in the early stages of development before the sponge is fully formed into a sponge. And then there are genes that are turned on quite late when the sponge is already forming an adult body type. And the genes that are turned on later in development are more like the ones that are turned on in normal development in other metazoans. So to give you an example of these, so here are 3 pathways that are found in the development of most metazoans that you go and look at. And early on in development, stage 1, low levels of expression of these canonical genes. And the further you go on towards later stages, the more canonical pathways you find, at least activated. So the genes that are turned on early in development are odd and weird and sponge-specific. The genes that are turned on later are more like those that you'll find in other metazoans. So in terms of those 10,000 weird genes that I was talking about in the 40,000, those will tend to be sponge-specific, Ephydatia-specific, and have roles specifically in doing sponge things. So the other thing we need to keep in mind is Ephydatia is not representative of every sponge. It is a freshwater sponge. Freshwater sponges are one peculiar lineage of sponge. What is in the genome of Ephydatia that's not in the genome of other sponges that enables it to live and survive what is a quite different environment to the sea? So something I'll mention very quickly, and it won't go into the depth of these slides, these graphs, is that in terms of gene losses and gains, Ephydatia, which is here, has a higher level of loss and also a sponge-wide shared signal of gain that indicates that gene gain is probably an important feature of all these new genes. So these are genes that don't exist outside the sponges. They're doing sponge-specific things. So a lot of these genes can be traced to the base of freshwater sponges. And so all of this novelty is feeding into their ability to survive in quite new conditions, almost certainly. So what kind of genes are we looking at? So we were able to look into a couple of different gene families because it's quite clear which gene families are replicating. Here's an example of one of these, GABA receptors. So GABA receptors are involved in homeostasis and control of response to stimuli. Homeostasis is obviously going to be different in freshwater to how it is in seawater. And we see a lot of tandem duplication going on here. So each of these dots is a tandem duplicate. You can see that they're arranged in little clusters. And over here, you can see the phylogeny of these. Any of these, This is quite hard to read, I realize, but any of these with quite a short name is Ephydatia. Freshwater demo sponges are going nuts with their GABA receptors. They're just duplicating like crazy. Ephydatia is no exception. So these are obviously really useful genes to have in a freshwater environment. Here's another example. Aquaporins and aquaglyceroporins, as the name suggests, involved in solute exchange. There are numerous copies in the Ephydatia genome. Here they're indicated with green dots. All of these are freshwater sponges. Freshwater sponges tend to have several copies. Other species tend to only have one of these. So having more copies gives you more ability to come up with new evolutionary mechanisms to control things. And obviously this is important. Here's one last example, the TRP or transient receptor potential channel families. So these are expressed around cilia and Ephydatia, which you can see from this fluorescent micrograph. These are, again, very highly duplicated in Ephydatia. And there's many copies in sponges in general, but Ephydatia is no exception. So you can see lots and lots of copies. So the kind of genes that are important for moving into freshwater environment tend to be things involved in transport across membranes and maintaining osmosis as you'd like it to be. So another thing we were able to do is compare across the gene families that we looked at, tests for positive selection of genes. So there's lots of different tests for selection. You may have heard of some of them, DN over DS tests. There's a few more sophisticated ways now. These are multiple tests, and you've always got to be a little bit suspicious with them that you're testing again and again, so there may be some false positives. The way around this is to do all the tests on all the genes and look at the ones that always give you a positive result. The consilience gives you confidence that these genes do have real signals for positive selection. And while not all tests pinpointed the same locations on the same genes, they generally agree on the genes that are under the strongest selective pressure, which tend to be things that are involved, again, in membrane transport, signaling and things like that. So what does this genome tell us about the peculiarities of freshwater evolution? Gene duplication is prevalent. Lots of genes that are duplicating are involved in sensing and responding to the environment. And there are some genes, a subset of these genes, are under quite profound selective pressure. So in conclusion, just to go over briefly what I've talked to you about, hopefully I've convinced you that this is a very high quality genome assembly for a sponge in particular, that it gives us insight into animal-wide characters that help tell us why animals are so special, but also allows us to make inference about the peculiar characters of Ephydatia itself and its related freshwater sponges. Not many things have moved into freshwater over the millions of years of evolution. If you think of the phyla that are found in freshwater, there are some mussels, there are some arthropods, there are some vertebrate, but there's not the 30-something phyla that you find in saltwater. So freshwater adaptation is special. This is a great way of looking at that. And just finally, Ephydatia in general is a great lab model. So if you wanted to use a sponge to study in the lab, there's no sponge that I can think of that is easier to work with than Ephydatia itself. So if I can finish with two quick ads. The first is for this resource itself. Please go to Ephibase. We've got lots of extra information that I haven't talked about today. And the paper that this is all based on was published about a year ago now. But you can go and grab that off that link there. And one very last ad for what I'm actually going to be doing in the last 5 years. So next 5 years. I've just started a Rutherford Discovery Fellowship at the University of Otago in the Biochem department, but working with the Cawthron. So I'm actually in the Cawthron Institute with all the nice people here in Nelson as we speak. So I will be looking at kuku, green-lip mussels or green shell mussels or kutai, whatever you want to call them, and trying to figure out why some lineages are more able to cope with the pernicious aspects of climate change. So things like ocean acidification and rising temperatures. Some kutai, some kuku are able to survive, some are not. What is the thing about the genomes of those who are resilient that makes them more resilient? And that's really important for both looking after those resources, kaitiakitanga, but also really useful for aquaculture, breeding in lines that you want to survive all these things. So I'll be working on that for 5 years. If you're interested in that, I'd love to hear from you. Yeah, hopefully I'll get to bump into you somewhere very soon in a COVID-free New Zealand. So last but not least, I need to thank a lot of people, in particular Anna Riesco, who was my supervisor while I was doing this work at the Natural History Museum in London, and Sally Lays, who was the instigator for all this work, the goddess of sponges, who's over at the University of Alberta, who gave me the samples to be working with and helped pay for the sequencing, which is very important. So if you've got any questions, I'd love to hear them. And thank you very much for coming along today. Cheers.